Hi there. Welcome to the world of biology. So, what exactly is biology? It's basically the scientific study of living things. We study everything from animals and plants, bacteria, archaea, protozoans. In order for something to be considered living, it has to have one of the 7 properties of life. But how did scientists kind of figure out these properties of life? They looked at unity and they looked at diversity. So in unity, we know that different organisms of different classes have commonalities, such as birds all have wings and feathers, or humans, primates all have stand upright and have a nervous system and a large brain. All of these different common sets of characteristics are all based on how organisms have evolved, and they've evolved from each other. Another property of life that's very important is the diversity of it. Because there are so many different environments and places that all of these different organisms live, these environments have different requirements for those living organisms to survive. So organisms have evolved different ways to survive in different environments, and that evolution of those organisms has provided diversity. So what are these characteristics of a living organisms? Scientists kind of all came together at some point in time and decided there were 7 characteristics that were required for something to be considered living. They had to be made up of cells, and the cells had to be organized. Now, you may say, well, a paramecium is only a single cell, and that is true, but that single cell is well organized. It has to be able to use energy and have some form of metabolism in order to derive that energy. And we're going to spend Unit 3, looking at metabolism and the properties of metabolism. They have to be able to respond to changes in their environment. You get cold, you put on a jacket. You get warm, you take the jacket off. So you are responding to some type of change in your environment. There must be regulation and homeostasis. An organism has to be able to regulate its own biological processes. An example of this would be we maintain a body temperature, so that's homeostasis. Our bloodstream maintains a buffer system, a bicarbonate buffer system, so that our blood doesn't become too acidic or alkaline. So we have all different forms of homeostasis, and we have to be able to regulate. Cells have to be able to regulate their own processes in order to maintain that homeostasis. Unit 2, we're going to study a lot of the processes that are used by human cells, eukaryotic cells, for regulation. Organisms have to be able to grow, and throughout that growth, develop. We think of growth and development in humans, such as we are born as these infants, and as we get older, we get stronger and bigger, so we increase in volume, we grow, but we also develop, and the organism becomes wiser and learns from its environment and incorporates other things, and develops physically as well. Reproduction. A living organism has to be able to reproduce. What form of reproduction is not as important, such as asexual versus sexual or budding, those sorts of things, but it has to be able to reproduce. And finally, the organism has to be able to evolve. Now, we're not looking at the evolution of a single individual organism, but we're talking about the species over time. So organisms of the same species, that species as its environment changes, as the world changes, and it will over time, those organisms, that species or animal or plant, has to be able to evolve over time in order to adapt to those new environments. So these are the 7 basic characteristics that are required for something to be considered living, or the characteristics of life. Here we have just from your textbook some examples. We see, of course, cells and organizations. So here we have a zebra, multicellular organism. The use of energy and metabolism. Photosynthesis is plant metabolism. Responding to environmental changes. This is phototropism in plants, where they're going to move towards the sunlight. We will actually learn about the plant hormone that is regulated by the absence or presence of sunlight, which is what causes these plants to move or bend towards light. An organism has to be able to regulate and maintain homeostasis. So here we have a bird that's maintaining body temperature in cold weather. Growth and development, organisms have to be able to grow and develop over time. We also have reproduction, and we have evolution, organisms changing or a species changing over the course of several generations or over the course of time in order to adapt to a changing environment. Organisms have different levels of organization. So when we talk about levels of organization, we're talking about from the molecular level to the biosphere level. So we start out with atoms. Atoms come together to make molecules and macromolecules. These molecules and macromolecules, come together to make cells. Cells make tissues. Tissues make organs. Organs make an organism, population to community to ecosystem, and then eventually to the biosphere. So we have these different levels of organization. Some examples of evolution are different changes, and one example is changes to what are referred to homologous structures. So we have a bat wing, which has 5 phalanges. We have a rat foot, which has 5 phalanges, and we have a dolphin fin that underneath the skin and blubber layer has 5 phalanges as well. They are all structurally similar. They serve similar purpose in motility for these organisms, but as you can see, they're in very different environments. One is a wing, one is a foot, and one is a flipper. So legs that were used for walking were eventually modified into the flipper of a dolphin or into the wing of a bat. When evolutionary change occurs, we have 2 very distinct types. We have vertical descent with mutation, and we're going to look at horizontal gene exchange. So we have vertical change and horizontal change. Now, in vertical descent with mutation, we see a change as a progression. So from parent to offspring. We inherit traits from our parents and we pass certain traits down to our children. So this is referred to as lineage. And in lineage, we see change in a vertical descent type of situation. In lineage, mutations. So there's some random changes that will occur in the genetics of an organism. These can be spontaneous mutations, so it can just occur randomly out of the blue. It's a really big mutation, and all of a sudden somebody who comes from a family where no one was over 5 foot is this anomaly and born over 6 foot, right? Becomes over 6 feet tall. These random mutations that occur can sometimes be beneficial depending on the environment in which the organism is in. So if a random evolutionary change or genetic change occurs into an organism and that mutation results in an organism that is now better able to survive an environment, then we're going to see over time an evolutionary change through vertical descent. Because now that organism has that mutation, it's going to pass that mutation on to its offspring. So when these mutations are beneficial to the organism in a changing environment, it increases the ability of this organism to survive. And if it has a greater survivability, it's going to have a greater chance of reproduction. In natural selection, a beneficial mutation causes an increase in a population. One evolutionary change through natural selection was horse teeth. Horses had very small teeth, but they had to widen the surface of the tooth in order to grind the grasses that they were eating and absorb the nutrients out of those grasses. So if you think of it, horses were actually very small forest-like creatures. They kind of maybe the size of a fox or a small dog. And then over time, as they moved into these prairie areas and needed to be in more wide open areas and got bigger, they started to develop teeth that were flatter and much larger, and those teeth were better able to grind the grasses that they needed in order to survive. Now, in horizontal gene transfer, the second type of evolutionary change, This most often is going to occur in a prokaryotic organism. Uh, Prokaryotes include bacteria and archaea, and these organisms directly transfer genetic material. So there's no offspring. You start out with two cells and you end with two cells. These are two cells that are just exchanging genetic material. There's no sexual reproduction. Now, they don't pass on their genome, they pass on pieces of their genome. And they don't really pass them on, they just swap between them. There are three forms of horizontal gene transfer. We have transformation, conjugation, and transduction. All three of these occur in bacteria. In transformation, bacteria come into contact with DNA that's just randomly in the environment, probably from another bacteria or cell that has died and broken down, and the DNA is left behind. So a living bacterial cell will take up that DNA and transform itself using the new DNA that it took up. Conjugation is when two bacterial cells come into physical contact with each other with a specialized structure called a pilus, and they will literally pass DNA through the pilus. It's like a big hollow tube, and they'll pass DNA through the pilus to each other so that one cell will receive new DNA and will incorporate it into its genome. The last form is called transduction, and in transduction, bacteria get sick to viruses. So viruses are considered non-living, and these little entities, there's a special class of viruses that can attack bacteria, and they're called bacteriophages. And these phages can take up DNA from one bacterial cell, and when they go to infect another, drop that genetic material off, and that host cell will then incorporate that DNA into its own. This direct transfer of genetic material can occur between the same species of bacteria, and sometimes it can occur between different species of bacteria. Some examples of genes that get transferred are antibiotic resistance, toxin production, the ability of a bacteria to produce a capsule, which is kind of like a protective outer coating. They can pass on dissimilation factors, which are odd metabolisms, allowing organisms to survive in new environments. When we classify organisms, this classification is called taxonomy, and organisms can be grouped, or species can be grouped based on their vertical descent and what we call homology, or common characteristics. There are three primary domains of organisms. We have the archaea, which are prokaryotic organisms, the bacteria, which are also prokaryotic, and the eukarya, which is your plants and animals and pretty much everything else. Each domain is then further broken down into kingdom, phyla, class, order, family, genus, species. And in biology, we use what's known as binomial nomenclature, which is the genus species method of naming different organisms. We do need to know the difference between a prokaryotic and a eukaryotic cell. Prokaryotic cells have very simple cell structures. They have no true nucleus. They do not have organelles that have their own membrane or envelopes. And prokaryotes take part in vertical and horizontal gene transfer. Eukaryotic organisms have very complex structures. They have nuclei, so they have a true membrane-bound nucleus. They also have membrane-bound organelles, and those organelles all work autonomously as well as together. And then eukaryotes evolve primarily through vertical gene transfer. When looking at evolution and changes in organisms, we have to know the difference or understand the difference between a genome and a proteome. The genome is the complete makeup of an organism in its genetic form. So all of its DNA, all of its genes, all of its chromosomes, all of that is its genome. The relatedness of organisms genetically is referred to as genomics, and when it's written down on paper and they kind of make these big genetic trees, it's a form of comparison known as cladistics. So in cladistics, we compare genomes and we look at percentages of similarity, how similar is the genetic code between one organism A and organism B. Proteomes is the complete complement of proteins that are the result of a genome. Genes in a genome, so an actual gene of an organism, if it's a structural gene, it encodes for a protein. And proteins do all of the work in the cell. So when we study genetics, we also have to consider the products of those genes. And the products of those genes are proteins. And the entire makeup, your entire protein makeup, is referred to as your proteome. So the genome is what carries the information to make all of the proteome, and the proteome is what really makes you, you. So all of your proteins together are what make your cells function. Your DNA is just simply the blueprint for it. Now let's talk about biology as a discipline because it's very important. In science, we observe, we have to identify different things. We investigate things by experimenting, comparing, and testing, doing things like that. And then eventually, based on what we tested, we come up with some kind of idea of the purpose or which is better and which isn't. And we make these kind of very educated guesses and we draw some conclusions based on the evidence that we have come up with through our experimentation. Ultimately, science is the theoretical explanation of a natural phenomena. So we observe something, we see something really interesting and such, and we go through this scientific process in order to identify what's actually happening. We investigate life at all different levels. These are all different fields of biology or branches of biology, you could say. These guys, we have ecologists and A&P people, cell biology, molecular biology, systemics biology. We have biotechnology and recombinant DNA and geneticists. There's a million different fields of science that you can go into. The point of this, however, is that all of these different fields all use the same investigative process, which is the scientific method. So, in the scientific method, once you observe some type of phenomenon that you want an answer to, the first thing you have to do is develop a hypothesis. Now, a hypothesis is just simply an educated guess. We're trying to explain something or make a prediction of some kind. An example of a hypothesis is that maple trees drop their leaves in autumn because of shortened amounts of sunlight. So we're thinking, well, sunlight is necessary for the photosynthesis to occur, and in fall, these trees are going dormant. They're not going to be carrying out photosynthesis. They're losing their chlorophyll. As daylight must play a role in this. Do we know that for sure? Not until we test it and we come up with a way to determine if sunlight plays a role. Now, a theory is a very broad explanation. It has the idea that the natural world, some aspect or some phenomenon that occurs in the world is actually able to be not necessarily proven, but it's able to be trusted, and that's because there's so much evidence through tons of different experiments and other scientists that we universally accept a theory as truth. Now let's look at how we develop a hypothesis using scientific inquiry. A scientific hypothesis has to have two very important qualities, and the difference between these two qualities actually is almost non-existent. So it must be testable, which means it must be falsifiable. Falsifiable just simply means testable. It's just a fancy way of saying we have to be able to prove something wrong and not necessarily go with we have to prove something right. If something is not falsifiable, then we would always still prove it right. So we have to be able to prove it wrong in order for it to be tested. Now, data collection from the testing of a hypothesis can also be collected in two different forms: quantitative, which is mathematical or some type of measurement, or it can be qualitative, where we describe it. Examples of these would be if you're running a fever. If you're running a fever, quantitative measurement of that piece of data would be to take your temperature with a thermometer. A qualitative description or a qualitative data point would say their forehead felt very hot. You would just feel it. So qualitative, think quality; quantitative, think quantity. What is the point of a hypothesis? Well, it's to make a prediction. And remember, you have your hypothesis, or what's referred to as an alternative hypothesis, and your null hypothesis. And interestingly enough, the null hypothesis is actually what you're testing. Because remember, your hypothesis has to be falsifiable. So ultimately, when you gather all of your data and you analyze all your data, and there's some kind of statistic that you may use to compare two different things. You either accept your null hypothesis, which is this affects this, or you reject it. If you reject the null hypothesis, then this means you are providing an alternative. So when you develop a hypothesis, there's really two. There's the null hypothesis, what you're actually testing, and there's the alternative hypothesis, which is what you accept if your experiment shows that your conclusion was incorrect. I know it sounds a little confusing, but always remember your null hypothesis is the one that you are testing. The alternative is what you accept if your null is not true. Other observations or experiments are needed in order to support or reject your hypothesis. That's why there's usually some kind of statistic or number that you're going to be looking for. And a hypothesis is never proven. It can be disproven. We can show that something does not happen, but we can never really 100% show that something does. We have to always assume that we found an explanation, but maybe it's not the whole explanation. There may be more to it. So we do have to accept the idea that a hypothesis is never actually going to be proven 100%. A theory is a really broad explanation, and we can actually make lots of guesses about the world around us based on different theories. Right? So we also can never totally prove a theory to be true, but it's majority of us, we accept it to be true. So the theory that DNA is the genetic material. No one has provided any information otherwise. Everything points to DNA as being the genetic material. We universally accept that. We still can't always say with 100% certainty that it is. Now, two key attributes of a theory is that it has to be consistent with a large amount of data that's known, and it has to have the ability to be used to make many different correct predictions. Now, when we're going through this hypothesis-based science, more often than not, we use what's called deductive reasoning. And in deductive reasoning, we're going to go from the general to the very specific. So this is sometimes called a top-down approach. So we go from a theory to a hypothesis to an observation to a confirmation. And an example of that is a little statement here about watch out for that wasp, it may sting you. So we're looking at, in general, that all wasps must sting, to the very specific. That wasp might sting you. So if our hypothesis is correct, then we can actually predict and expect a very specific outcome. With inductive reasoning, our logic begins to move from the very specific to the more general. And we got to be a little careful with this one. If we use inductive reasoning in our scientific processes, we may be looking at too narrow of a spectrum, and we may be concentrating so hard on that narrow spectrum that we avoid or we prevent ourselves from seeing other factors or variables. So inductive reasoning, although it can be used, this bottom-up reasoning technique, we need to be a little careful with it. In order to understand biology, you've got to be curious, because the scientific method is not this really rigid, do it exactly this way kind of steps. The scientific method is a process, right? It's a process. And when we're ready to start using the scientific method, there are two different ways that we can approach it. We first have what's called discovery-based science, where we're not really sure what we're looking for until we find it. Then we have hypothesis testing, and that's where we come up with something very specific and we try to prove or disprove it. These are null and alternative hypotheses. So in discovery-based science, we're going to collect a whole bunch of information. We're just going to go out and start taking, I don't know, water temperatures in the ocean in different areas or something, just because we're curious what's out there. Once we start collecting this data, it isn't until we start putting it together that we start to see something happening. And that is so where we go from the really broad to the really narrow. So the goal of discovery-based science is just collect information. Sometimes you're not really sure what it is you're looking for, but you know you're looking for something. So you go to the field, or you go out to collect information, and you just collect as much different information as you can. In hypothesis testing, or in the scientific method, we are looking for something very specific. So there are 5 stages to the scientific method. The first is we observe, and we are looking for something strange or some kind of natural phenomena. Once we have these observations, we start to develop a hypothesis. So we're using this hypothesis to try to explain something that's happening. Once we've developed a hypothesis, we're ready for experimentation. So we go through experimental design, we design an experiment, we carry out the experiment, we collect all our data. All of this collected data is then analyzed, and the analysis of that data allows us to draw a conclusion and either accept or reject our original hypothesis. Now, data is usually collected in 2 ways. A control and an experimental sample, so these are kind of all known. And the control and experimental sample, the only thing we're going to have is one single factor of difference between them. So that's referred to as the variable. For example, if you wanted to see if the amount of sunlight actually did affect plants and plant growth, you would take 2 plants of the same age, of the same height, very similar in structure, as close as you can get, put them both in pots with the exact same amount of soil and water and all of that, and the only thing you would do is cover one for part of the day, so that the only variable or difference between them was the amount of sunlight they were getting. When we analyze data, we have to apply statistics in biology and biological papers and that sort of thing. Statistical significance. And statistical significance is done through, of course, statistical calculations, and we apply stats analysis in order to determine whether or not our samples are really that different from each other. Is my experimental sample different enough from the control sample so that I can safely predict that our variable is what has that particular effect? If there's no statistical significance in the difference between the two samples, then we would reject our hypothesis. If there is a statistical significance, then we would accept it. Now, here we have a case study in cystic fibrosis. This is from your textbook. You have a copy of this in the PowerPoint PDF file. So I am going to let you guys read the CF gene and hypothesis theory on your own, and we'll talk about it a little bit in class. So have a good day, and I will see all of you in class.